We are looking at mammalian osteology, and this is a continuation of the previous lecture that we had with birds and marsupials. And in this case, we're going to look at eutherian mammals, or in other words, placental mammals. And so the first one we're going to look at is an herbivore, a rabbit. And just to kind of give a little preview of this, we are looking at some similarities with the opossum. And specifically, what we notice is a plantigrade posture of the hind limbs. And plantigrade means that the whole foot is touching the ground. And then some differences would be the angles that you see here. You can see a very acute angle there in the knee joint. And that's, of course, because of the way that this rabbit gets around would be through hopping. And so having that angle is going to allow for more potential energy for movement with that type of locomotion. The individual bones, just an overview of this, we want to start with the scapula, and we want to look at how this scapula looks different from the one that the opossum had, which was a bit more broad in shape, and this one's a bit skinnier and more triangular in shape. Here we have the scapula articulating with the humerus, and so this would be the shoulder joint. The humerus articulates with the radius and ulna, and so then this would be the elbow joint, and then we would have a set of carpals and metacarpals going into phalanges. On the lower limb, we've got the 3 hip bones, and if you've taken human anatomy, you know that we've got the ilium, we've got the ischium, and we've got the pubis. And so those are there for the rabbit as well. And that's making up a one fused os coxae bone. And the acetabulum will be the socket where the femur can articulate in the hip joint. And then moving down over here, you've got the femur articulating with the tibia and the fibula, and that therefore is the knee joint. And then we've got the tarsals down here, which means this is the ankle joint, the metatarsals, and then the phalanges after that. If we want to look at the vertebrae, we can start with the caudal vertebrae, and these caudal vertebrae are making up the tail. And then once we get to an area that's close to the os coxae, that's going to be in between the 2 hip bones, we've got fused vertebrae, and that's known as the sacrum. So these are going to be between the 2 hip bones. And then once they unfuse, then we get to the lumbar vertebrae, which would be these right here. And as soon as we see ribs, we know that we're in the thoracic vertebrae section. And so the thoracic vertebrae, the way to define them is that if you see a rib attached, you know they are thoracic vertebrae. And for the ribs, you can see that we've got vertebral ribs, which are articulating with the vertebrae, but those also articulate with sternal ribs that articulate with the sternum or the breastbone. And if we move up, we can go to the neck region once we're past the ribs, and those would be called the cervical vertebrae. And for the cervical vertebrae, very similar to what we have in humans, we've got the axis as C2 or cervical vertebrae 2. And right above that, we've got the atlas, which is cervical vertebrae 1. And just remember that the atlas is the one that holds the skull. And this comes from the mythological person known as Atlas, who made a god angry in ancient Greece, and his punishment was to hold the earth on his shoulders for eternity. We call axis axis because it will allow for Atlas to rotate over it so that the animal can turn its head side to side. Now, if we look at this picture, this is a good practice slide so that you can hide the key down here and try to label these and see if you're labeling them correctly. And just make sure you go off your list of terms to make sure that you know which features you'll be tested on and which ones you don't have to worry about so much. But what this is good for, just a few things on this picture that does a good job compared to the other one, is that it really shows nicely how Atlas and axis are shaped. So you can really see Atlas right there and axis right there. So you can really see that they're very different from the rest of the cervical vertebrae that make the rest of the neck. And also what's nice about this is that you can see how those will lead into the occipital bone, which is going to be right after that. And then after the occipital bone, you would have the parietal bone, and then going into the frontal bone, and etc. We're going to have skull pictures coming up, so we shouldn't spend too much time here. But then here we have a nice zygomatic arch as well. What we notice immediately is that we have a set of incisors in the front, and we have premolars and molars in the back. And in between, we have a gap of no teeth, and this is called a diastema, which is a characteristic morphological feature in herbivores. And that is because what they're doing with their food is that they're slicing their food with their incisors and then grinding that food with the molars and premolars. And this in between space divides these two functions between the front and the back. And also what they can do is pinch their cheeks in, the muscular parts of their cheeks, and use those muscles to basically separate the two parts of the mouth, the front and the back, and also maybe squeeze out certain parts of their food that they don't really want to send to the back of their mouth, and divide and conquer in terms of their function of slicing and grinding front to back. The other thing that we notice is that we've got this nasal bone, which we can really see much better from the top view. And it's a pretty big nasal bone. Moving posteriorly, we've got the frontal bone, which is going to be found in between the eye sockets. The parietal bone after that, and then finally the occipital bone. And then after that, you're going to have the cervical vertebrae. From the top view, you can really see the nasal bone being very, very large, which tells you that these animals are using their nose quite a bit, and olfaction is going to be a big deal. You can also see from the side view this large external auditory meatus, which is the tube that allows for sound to travel to the tympanic membrane. And being so large, that also tells you that hearing is going to be another very important sensory feature for these animals, for them to monitor their surroundings. So looking at the top view again, you can really see how the nasal bone, if we just follow it backwards, is going to run right into that frontal bone. And the frontal bone is going to run right into the parietal bone. And the parietal bone is going to run into the occipital bone. If we look at this zygomatic arch here, we see that the anterior portion of the zygomatic arch is being composed by the jugal bone. And we've seen the jugal bone before, and just realize that in mammals, the jugal bone is often called the zygomatic bone. So you can use these terms interchangeably once we get to mammals. Either one would be correct on the test. The zygomatic or jugal bone will run into the squamosal bone somewhere in the middle here of that zygomatic arch and form the arch. And just remember that the squamosal in mammals is also referred to as the temporal. So you can again call these one or the other in terms of the test, and you would be correct for either one that you put down. The premaxilla is going to have the incisors. And so you can see these incisors here in the front. And so the bone that would give rise to them is going to be called the premaxilla. The maxilla will be right after that. And the maxilla will have most of the upper jaw and will have the teeth in the back, the premolars and the molars. And you can see that the jugal bone is going to be right above that. On the bottom, you've got the mandible, which is also known as the dentary. So again, either one of these names would be fine. When it comes to mammals, we start calling it the mandible. And here again, you're going to have all the teeth inside this bone. So the incisors in the front from the dentary, but also the premolars and the molars in the back. In this picture, we want to look at the dentition of the rabbit. And what we notice is that we have an incisor in the front called the first incisor, and an incisor right behind it called the second incisor, or sometimes known as the peg tooth because it's small. In terms of right after the diastema, we've got 3 premolars and 3 molars on the top jaw. If we compare that to the side view, we can see the first incisor being much larger and the second incisor being smaller. So this is a big characteristic of rabbits. And the premolars being 1, 2, and 3, and the molars being 1, 2, and 3. When we think about the difference of mammalian dentition compared to reptiles or fish, what we notice is that these teeth look different from each other. And we call this characteristic heterodont, meaning different heterodont teeth. In other words, the incisors look different than the canines, look different than the premolars, molars. But the premolars and the molars do look rather similar to each other. So the way that we differentiate between premolars and molars is that with mammals, we do have a set of milk teeth during our early years when we're breastfeeding. And then these teeth fall out and then are replaced by adult teeth. And so even though premolars and molars look similar, and it's sometimes hard to tell the difference, the premolars end up erupting out from what is in front of them, the milk teeth. Whereas when molars erupt, there's no tooth that is in front of it. And so it's just one round of teeth erupting without another set that follow them. From the side view, we also want to notice the eye socket position being very much pointing to the side of the animal. And this reminds us that these are, first of all, herbivores, but also the fact that being herbivores, they also end up becoming the prey of carnivorous predators. And so they need to have their vision to the side so that if you're on the side of this rabbit, they can see you rather well. And in fact, what they want to do is they want to be able to see their surroundings as much as possible and be able to scan their surroundings for any little movement. Also notice again a review, that big auditory meatus, which means they'll be listening for predators. And then, of course, they'll be using olfaction in order to smell their surroundings, in order to try to uncover different scents of different animals that have been there before in that surrounding or are there at that same time. The last thing we want to do here is look at the lower jaw, the mandible, and what we notice is that there's no peg tooth. So there's only one incisor on each side, and you don't have that second incisor right behind it. The other thing that we see are that we have 2 premolars and we have 3 molars. So the dentition number is somewhat different for the upper jaw compared to the lower jaw. Moving on, we want to look at the skeleton of a bat. And the reason why this is so fascinating is, of course, because bats are mammals who have figured out how to fly, and birds are famous for flight, but mammals have done a pretty good job as well. However, this mechanism of flight has been figured out on different occasions evolutionarily. And so this is an example of convergent evolution, which means that the anatomy will not be exactly the same, but will end up having a similar function. And so we do have wings, but these wings are built rather differently. If you recall from the bird osteology chapter, you had fewer carpals than 5, and the carpals seem to be more in a series, one on top of another, in order to extend the wing out so that those flight feathers could fan out more appropriately. But we don't have feathers in mammals. And so what we're going to use instead of feathers are flaps of skin that are going to be going in between these digits. And in order for this to be effective, what we need to do is we need to lengthen the digit significantly. And so you can see that from the humerus, you've got this radius ulna that is pretty long, that starts this wing structure. And then you can see that the phalanges, now this one is phalanx number 5, which tells you, if you're looking at your own hand, that's your pinky phalanx. And then if you try to put your hand in this conformation with the pinky up, your thumb ends up pointing down. This one right here must be phalanx 1, which means that this one right here must be phalanx 2, and this one 3, and this one 4, all the way to 5. And so again, this is 1 would be homologous to your thumb, 2 your index, 3 middle finger, 4 is the ring finger, and 5 is the pinky finger. Then of course you got the metacarpals that also fan this out, and then giving rise to the carpals right in there. Bats are fascinating because they're not very well understood, and there are a lot of stereotypes that go along with them. First of all, what is surprising is that 20% of mammal species are bats. So that's a huge number that most don't realize. There are, right now, counted 1,200 different species of bats. Now, we have these 2 broad categories. One category is called the mega bat. And these are the bats that are larger in size, that are typically going to be eating plant material like fruits. So they're called the fruit-eating bats. The second type are called the microbats, and these are the smaller ones who are also famous for their ability to see with echolocation. In other words, using sound to bounce off objects in order to locate those objects. Now, within these microbats, and again, most of them are going to be insectivores or maybe carnivores eating smaller organisms, but there are three species of bats called vampire bats. And these vampire bats are native to the Americas. And in fact, it's interesting if you read up on the history of vampires and Dracula and those types of vampires, you realize that those stories and their association with bats came about after the Europeans colonized the Americas and realized that there were these bats in the Americas that would basically puncture the skin of larger vertebrates at night, because these are nocturnal, so while they're sleeping, they would puncture their skin and let the blood drip into their mouths. So it's interesting to look at the skulls of these two types of bats. We've got at the top here this vampire bat, and what you notice here are these incisors look like they would do a really good job at making a little puncture in the skin of a larger vertebrate while that vertebrate is sleeping, and in a way that would not awaken that vertebrate, obviously. So we have to be very precise and a very small incision. And also what you notice is when you look at the eye sockets, they are very much sunken into the skull. And that's because these eyes seem to be much smaller than the eyes of a megabat, and that's of course because they're using echolocation, and you can really see that extremely large external auditory meatus, because if you're going to be using echolocation, you need to have a very big sound hole for the sound waves to be able to go in. The picture on the bottom, on the other hand, shows us the megabat. And here with the megabat, these are fruit bats, so these are not vampire bats. And what we notice here is this very large, big eye socket. So it's not true, for example, that all bats are echolocating, right? And so these megabats, the larger ones, are using their eyesight to see, and their dentition is pretty pretty standard. In other words, in the sense that they have incisors, then they have canines, then they have the premolars and molars that are going to be right after them. We're going to see that this type of dentition looks pretty similar to the dentition that you'll find also in cats. Let's finish this lecture of mammalian osteology with cats. And cats are a fascinating group, which include the large cats and the small cats, the domestic cats. And here what we've got is the skeleton of a lion. And you can see, for example, there's a lion with that mane right there. We should notice that these are digitigrade animals. And we can tell that they are digitigrade because if you look at the paws here, you've got in the hind limbs, for example, you've got the phalanges making contact with the ground, but the metatarsals being elevated, the tarsals being elevated as well. You do have 4 phalanges on the hind limbs. And then in the forelimbs, you do have 5. You have this 5th one right there that you can see. And so you've got those metacarpals that are lifted off the ground, the carpals lifted off the ground. And this tells you that these animals are built for speed. And what we need to do in order to make them faster is extend the limb to make the limb longer. Because the longer the limb, the longer the stride of the animal. So if you were to put this wrist down on the substrate, you would immediately notice that the length of this limb would decrease. And that would mean that for every stride, there would be less distance traveled. This skull is the skull of a domestic cat. And what we notice here, let's look at the dentition first. These are clearly going to be carnivores. The eye sockets are pointing straight forward, giving this animal binocular vision, which means they can really see right in front of them very well, and they can also tell the depth of what they're looking at. So they can know exactly how close or how far away something is from them when they have their eyes pointing forward. In terms of incisors, there are 3 on each side, followed by a massive canine. After the canine, we have a very small first premolar, followed by a second premolar, a third premolar, and then actually in the back, which you cannot see in this picture, but you'll see in future pictures, is going to be a small molar in the back. Now, this picture really shows you well how that premaxilla is giving rise to those incisors right there. So the premaxilla is where the incisors originate. And then right after that premaxilla, you have the maxilla, which is going to house the rest of those teeth. Then if you follow your way up, you'll see that you have the nasal bone making up the nose of the animal. And the nasal bone, then, if you follow it up, will run into the frontal bone. And again, the frontal bone, if you want to orient yourself, is typically going to be found in between the eye sockets. If we continue up, we're going to run into the parietal bone, and behind it would be the occipital. Now, looking at the side view and looking at this zygomatic arch right here, we know that this first bone that's making up that zygomatic arch will have to be the jugal bone, or also known as the zygomatic bone. And that second bone that makes its way back this way, and they're going to meet somewhere in the middle, is going to be the squamosal or the temporal bone. So here you can really see that. You can see how that jugal bone or zygomatic bone is making up the anterior part of that arch. And then you could see that this bone that is more posterior, the squamosal or temporal bone, is making up the back part of that arch. Now the squamosal bone is also going to give rise, because the squamosal bone is kind of complex and does have different parts to it. So it's also going to give rise to that external auditory meatus. And then in cats, as well as canines, you've got this bulla, this auditory bulla, which is this big circular structure at the base of the ear, which is going to be extra support for that middle ear where hearing is taking place. Now, moving more posteriorly, what we find are the occipital bones, and there are going to be a few different ones, but for this course, just knowing the occipital is adequate. And then what we're going to see is the parietal bone, and again, the frontal bone between the eye sockets, moving into the maxilla, the nasal bone, and then the premaxilla, which is going to have those incisors in there. For the mandible, or otherwise known as the dentary, that's going to house all of the lower jaw teeth. If we look at the bottom view here, what we can see is that premaxilla bone, and we can see those 3 incisors that are coming out of that premaxilla. And then after that, you've got the maxilla with the rest of the teeth, starting with that big canine, that small first premolar, the second premolar, the third premolar, and then 1 molar in the back right there. Further down, what you want to look at are these bulla, these auditory bulla, or sometimes they're called tympanic bulla, which are going to be these very round structures where the auditory meatus is found. That's where you're going to find the bulla. And also from the bottom view, you can see that this right here would be the jugal bone. This right here would be the squamosal bone, and they would meet in the middle, forming that zygomatic arch. And just so that we're reminded, all of these animals that we're looking at are synapsids, which means that they are going to have this singular temporal fenestra, or hole in their skull, where muscles will pass through. Here what we have is a very good picture of a cat skull in the lateral view, and it really shows us and labels for us a very good maxilla with the most of the teeth that are coming out of that maxilla. It shows you that jugal bone with the zygomatic arch being formed by the jugal and that sharp part of that squamosal bone. We can also see the external auditory meatus, the tympanic or auditory bulla, which is again this big round structure that houses the middle ear. And what we see in the back is going to be, of course, the occipital, followed by the parietal, and then the frontal after that. From the posterior view, you can see the foramen magnum, which is where the spinal cord will come out, and you can see the occipital condyles, which is where that first vertebrae, C1, or in other words, atlas, will articulate. So the first cervical vertebrae will articulate with the occipital condyles. And then what also we see here is this sagittal crest, which is down the middle, and then this lambdoid ridge, which is making that lambda shape. If you're familiar with the Greek alphabet, the letter lambda, that's what it looks like. And it's basically where these bones meet. So you've got the parietals meeting together in the middle, and then you've got the occipitals meeting together as well with the parietals. And this is making this this kind of 3-directional suture structure that meets right at the middle, right there. Here, this is a very good picture of how large those tympanic bullae are. And you can see also that's labeled here is that palatine bone, and this is making up the hard palate. And then you can imagine here that as air is going through these internal nares right here, In the living cat, you would have a soft palate that would kind of create a shelf right here, which is, of course, we cannot see right now because this is just the bones, but the soft palate would be made up of soft tissue. And then we can really see that premaxilla giving rise to the incisors, the maxilla giving rise to the canines. There's the first premolar, the second premolar, the third premolar, and then the molar, the singular molar in the back. And again, looking at the jugal with the squamosal, the zygomatic arch, and the temporal fenestra, or opening right there, telling us that this is a synapsid. And when we compare this to a lion skull, Panthera leo, what we notice is that everything is pretty much the same. The only difference is that this skull protrudes more. I would say that the domestic cat skull is much more squunched up and doesn't elongate itself as much. But everything else is going to be the same. What we can really see here is very clearly that suture right there between the jugal and the squamosal. You can really see the maxilla and the premaxilla. And on top, you're going to find the nasal, the frontal, the parietal, and then back here, what you would find is the occipital. And on the bottom is, of course, the dentary or the mandible. And again, once you get to mammals, you can use either word interchangeably. This is a good picture to study from, and you can basically try to label this as you go. Again, look at your list of terms and make sure that you're not labeling things that you're not necessarily going to be tested on, unless, of course, you want to. And then over here, we can look at the lion dentition and see how similar it is to the domestic cat, with 3 incisors from the premaxilla, and then the maxilla giving us that big canine, that first premolar, that second premolar, that third premolar, and then that little molar in the back, which you can see is being labeled right there for you. And then here is that side view of the same thing. The lower jaw dentition is a little different. We still have those 3 incisors. We've got that canine right there. But in the back, what we notice is that we've got 2 premolars and 1 larger molar. And again, the way that we define premolars and molars has to do with milk teeth and adult teeth. And so you would not be able to tell them apart necessarily just by looking at them, but just knowing that premolars did erupt from behind milk teeth, but molars erupted by themselves without a milk tooth preceding it in and being right before it.